So yesterday's class was on plant cloning vectors, and I had told you that if you want to transfer a gene to a plant, then one of the ways that you clone your gene in the TI plasmid, either through binary vector strategy or through co-integration strategy, and then you put that particular these two vectors inside agrobacterium. You infect your plant cells, generally in the suspension form, with agrobacterium. So agrobacterium will be able to get inside the plant cell and transfer the gene. And then you can regenerate your gene from the plant cell, first by inducing callus, by having this high concentration of auxin and cytokinin, and then from the callus you can regenerate the shoots, and then the shoots by first giving cytokinin and then auxin, and then this is how you generate your transgenic plant. Then I had also told you that apart from agrobacterium based plant transformation, there are other methods also, and especially these will be useful for those plants which are not very much infectable by agrobacterium, because we discussed that agrobacterium does have host specificity. So in that context, is there some other way by which you can transfer? Yes, there are other ways possible, though still agrobacterium mediated transfer is considered the most easy method. Anyway, so one method is that it was seen that when you incubate your plants, generally you need to remove their cell walls. So if you incubate your plant protoplast in presence of your cloning, let's say recombinant vector, then that recombinant vector, I mean, how will you transform the protoplast with recombinant vector? I'll just come to that. But then once it gets into the protoplast, then this your gene can get integrated by non-homologous recombination. We still don't understand how exactly this recombination process takes place. But yes, it happens at some random sites, and your gene of interest, it goes inside the plant DNA. And then that's how you can make your transgenic plants. Now comes the question that how will you transform the plant protoplast with your DNA? So again, there are few methods. One is, let's say, chemical based, one is physical based. So there are two ways by which you can introduce the DNA into plant. So first step is that you take plant cells, you remove their cell wall. So there are certain enzymes available which can remove the cell wall. So what we get is a protoplast. Then you incubate this with your DNA, but then the entry of the DNA into the protoplast is much better if you have it in presence of, let's say, one method is PEG mediated. So you coat your DNA with PEG, and PEG helps in the internalization of the DNA inside the plant cell. Alternatively, there are other methods also. Some people are making nanoparticles. Then there is liposome, that is lipid mediated entry of DNA into the protoplast. Then a silica based method for entry of DNA into protoplast. So there are various ways by which you can promote the entry of DNA into the protoplast. Many of these are being used in various labs. So once your DNA gets inside the protoplast, then you regenerate the cell wall, and then what we get is a transformed plant cell, and then again through induction of callus, and then means adding cytokinin and auxin, you can regenerate the whole plant. So this is another method, and it is mainly used for those plants which are not very easily transfectable by this agrobacterium. But it means still in this case also we have certain limitations, because not all plant cells, they show good regeneration from the this from protoplast. Means it was seen that in some plants it was very nice and you can regenerate the whole plant from protoplast, while in others it was not that successful. So depending on which plant a lab is working on, they have to standardize which method works best for them. Then another method is biolistic, which I had mentioned briefly in my last class, and it is a method in which what you do is that you take your DNA and you coat it with this heavy metal such as gold or tungsten, and then you pass it under a lot of pressure, a very high velocity. So then at the end of this machine, you put a rupture disc. So then what happens that your DNA, which is coated with these heavy particles, it goes with a lot of pressure to this disc and it ruptures it. And these particles are then made into very small particles, and then they get into these plant cells. So you can plate your callus here. So many times you generate a callus here, and then you bombard the callus with this particle bombardment machine, and then some of the cells in your callus, they take up the DNA of interest, and then using various selection markers you can identify the cells which have taken up the gene, and then you regenerate the whole plant from that particular cell, like I had discussed previously. So this is another method. So one is agrobacterium, then you can use chemical based methods by coating your DNA with PEG or encapsulating them in liposomal particles or nanoparticles, silica particles, etc., and see their internalization in protoplast. Or you can bombard your callus or even sometimes the leaf section or some part of the plant with this particle gun, and the DNA with so much velocity sometimes gets inside the nucleus by penetrating first the cell wall and then getting into cytoplasm and finally nucleus. And once inside the nucleus, it does get a chance to get incorporated or integrated into the genome, and that's how you can regenerate finally your transgenic plant. Then the last method that is used in or being, let's say, investigated in certain plants is the use of plant viruses. So the use of, so it is similar like bacteriophages were used for transfer of genes in bacteria. So similarly, various plant viruses are being tested whether they can function as cloning vectors and transfer your gene of interest into plant cells. But do we have the ideal plant viruses as vectors yet? No. The two most commonly used ones are colimovirus vector, and another is Gemini virus. These two viruses are used as vectors for transfer of your gene of interest into the plant cell, but then they come with their own advantages and disadvantages. So because of the disadvantages, it is this means I want to say that in some plants there are success stories with both of these viruses, but still for other plants or other transformation of your gene of interest, people are still trying to improve these vectors to be used as cloning vectors. So let's see that which viruses are used. So generally the plant viruses, they are RNA viruses. So all these notes are here. So because they are RNA viruses, obviously they are not good for manipulation purposes, because you will prefer a DNA vector in which you can clone your gene of interest. And do we have many of the DNA viruses? No, not many, but yes, some DNA viruses, plant DNA viruses are there, and the two examples are given here. And the disadvantage of cloning in colimovirus is the size of the insert is not that big that you can transfer in, or let's say clone in these viruses, because the problem is of that head thing, that it has to get encapsulated within the head of the virus, and also it suffers from narrow host range. So this virus cannot equally infect all the different types of plants. It does have its particular host range. And similar is the case for Gemini virus. In this, the disadvantage is that once you have cloned your gene of interest, it was seen that the gene that has been cloned is not very stable. You see a lot of rearrangements or deletions. So, as a molecular biologist, you would try to do that. Let's say if you see that the infection with Colimo or Gemini virus is very good, so you feel that okay, this is a very good strategy for transfer of your gene of interest, but you find that these disadvantages are there. So then, what will you do? You will try to study its molecular biology in very detail. You will try to identify the genes which are responsible for example for narrow host range, or which are responsible for rearrangement, etc. Then you will try to manipulate those genes and then see that whether now your these viruses, they can function as better cloning vectors. So again, I would like to say that improvements are being made in these two viruses to make them as good cloning vectors. But again, I would like to say that the most commonly used one, the most easy is agrobacterium. You just infect your plant cells with those bacteria and the transfer is very efficient. It's only for the rest of the cases in those plants where it is not very successful that you resort to rest of the strategies that I mentioned. Okay, so that's not all for plant cloning vectors. This table gives you a list of the selection markers. So like for bacteria, or let's say the plasmid vectors or the yeast vectors, etc. I told you that there are certain selection markers. So similarly, you have selection markers here. And apart from the selection markers that you will commonly see in many plant cloning vectors, you also have various reporter genes which are there. So these are, for any cloning vector, you will always find a selection marker and a reporter gene. And with respect to the reporter gene, I'll just bring your notice to this GUS gene. So GUS assay is very commonly used for identifying the transformants. So GUS assay is based on this enzyme, beta glucuronidase, which converts X-gluc to finally give a blue color by certain oxidative dimerization reactions. Okay, so if this enzyme is being made in any cell and you give X-gluc from outside, then you will see blue color in the transformed cells. Okay. So this is one GUS assay that was done. So this is onion peel, and means this in the cloning vector, you had this 35S promoter. So this is one of the promoter that is used in the plant cells. And also I want to tell you that in many of the plant cloning vectors, like in the case of plasmid cloning vectors, if you remember that the one of the promoter that is very commonly used is T7 and sp6 promoters in case of plasmid cloning vectors, which are to be put in bacteria. And why these T7 and sp6, which are bacteriophage promoters, they were being used because they were very, very strong promoters, giving a very high expression and transcription from those promoters. So that's why they were preferred. So similarly, in case of various plant cloning vectors, what they have done is they have used many of these CAMB promoters, these colimovirus derived promoters. So because these are viral promoters and we know that virus divide very fast once they infect, so that's why these promoters are used in many of the plant cloning vectors. So anyway, so this is a promoter and we have this GUS gene clone downstream to it. So somehow if we are able to transfer this plant cloning vector or this gene of interest into the plant cell, so the cells which have taken up this plasmid, they will show blue color. So this is how it is seen. So now coming to the panel on the right. So here what they have done is that in because it's a reporter assay, right? So what they have done is that, let's say this is your gene of interest and you want to see that where it is localized inside the plant cell, whether it is nucleus localized, it is cytoplasmic, or is it membrane bound and all. So what I do is that I fuse it with the GUS reporter. So I think that in your these assignment lectures, they must have told you that what is the importance of these fusion genes that are made or fusion tags that are added. So one of that is also you can look for the localization of a gene. So in this case, what they have done is, this is my gene of interest. It has been fused with Gus under control of a 35S promoter. So what will happen, because my gene of interest was nuclear localized, so what you can see that in this case, the gene is showing expression only in the nucleus. So this is how you can look for the localization of your gene of interest. One of the ways, by cloning it under fusion as a fusion protein, either with Gus, or you can also use GFP. So the cells which have taken up this cloning, recombinant clone, they will fluoresce green if they have been fused with GFP. And if you see under high magnification, you can also see their localization within the cell, whether it is mitochondria bound or whatever, membrane or nucleus or something else. So that's all for the plant cloning vectors. And just very brief overview of the expression cloning vectors, in which I'll only stress upon the pet vectors. And why am I skipping some of the facts is because in many of the assignment lectures these have been covered. So expression vectors, how do they differ from cloning vectors? In only the fact that they do have the sequences which help in expression of your gene of interest. So to ensure that your gene of interest is transcribed and then translated, what all sequences are to be added in the cloning vectors to make it an expression vector? These are some of the points that are essential, or they somehow influence the expression of a particular protein. So what you need in your cloning vector is, of course, the presence of a promoter. Without the promoter, the DNA that you have cloned will not be transcribed and then translated. Another fact is that if your gene of interest has been cloned downstream of a promoter and it does not have a transcription termination site, even that is not good. So most of the commercial expression vectors, they also have at the end of the MCS. MCS is the region where you clone your gene of interest. So here is promoter, this is MCS, you clone your gene of interest, and at the end of it, what you have is the termination sequences. So depending on whether you are having a bacterial expression system or you are having a mammalian expression system, you will decide that which termination sites are present. So if you remember, in case of bacteria transcription termination, it happens either in a row dependent or independent fashion, and there is present of certain U at a stretch, etc. So these things are taken into account, that if you have a plant clone, this bacterial cloning vector, you will add certain termination sites at the end of the NCS. So whatever transcript is being made from this promoter, it gets terminated very, very efficiently. And how do they ensure that the termination is very efficient? By, in many commercial vectors, you will not only see one termination site, rather they will have two termination sites. So from where do they decide that which termination sites to be added? They are added on base of previous studies, various published studies, in which they identify that, okay, this termination site is very strong. So based on those research from basic research findings, they decide that, okay, let's put these termination sites. So if you are given a map of a particular expression vector, you please look for these termination sites also, which are present in those commercial vectors. So if it is a mammalian expression vector, you will see different termination sites, and plant may be different, and for bacteria it will be different. Then I'm sure that this has been taught in a lot of detail, the presence of ribosome binding site, because if your transcript is made but it is not translated, if you're looking, your final product is protein, then of course it's of no use. So you ensure while designing your primers also, or maybe in some commercial vectors you already see a Shine-Delgado sequence, the presence of these ribosome binding sites. So the sequence and all must have been told to you in the assignment lecture, so I'm not going into detail. Then many factors guide how efficient your translation will be. So I think these points I will revisit when I'll be telling you about mammalian expression vectors and how the presence of certain sequences at the start of a transcript, they affect the translation efficiency, and how by adding certain bacterial sequences as fusion tags at the start of the transcript, you ensure that the translation is comparatively more efficient as opposed to when those fusion tags belonging to the bacterial origin were not put. So some factors means the presence of certain sites in the transcript, they do affect the efficiency of translation, and now we know certain fusion tags which promote the efficiency of translation. So if for commercial purpose especially you are making your protein of interest in high quantity, you add those fusion tags. Then coming to obviously the factors which affect the high level expression of genes is also the intrinsic stability of the protein. So again, it was seen that if you add certain fusion tags, the stability of the protein was increased many folds. So what those fusion tags are, we'll discuss in my next class, which will likely be after your minors. Then also another factor, that if you want to harvest your protein in very high amount, then what factors can affect the final protein amount that you can harvest is, one thing is that it will be best if your host strain, it is able to secrete your protein of interest into the media. So that will be like the easiest form of harvesting the protein, rather than if let's say your protein is present within the cell, be it a yeast cell or a bacterial cell, and you have to harvest it, then you have to obviously first lyse the cells, and you have to protect your protein from various proteases which are present within the cellular environment while isolating it. So that comes as one negative point. So best would be that if somehow you add maybe a secretion tag in your construct and ensure that your protein is secreted out of the cell, and what you have to just do is harvest the media and the protein which is present in there. Then in many cases, it is also the copies of the clone gene which are important. So I'll forward one review article to you, and all these points are discussed in a lot of detail in that review article. It's not a very big review article, but a short review article. And finally, of course, it's the host organism. Can you engineer your host strain in such a way that you get very high level expression of your protein of interest? Yes, you can do that. And some of those strategies I had discussed when I told you about the host strains. Oh, I will skip these basic points because these have been covered in some of the assignments. Let me just, like I told you, that which promoters you use when you're talking about uh, yeast expression. So some of the promoters I told you yesterday. So these are the list of promoters which are commonly used when you want to express your protein in bacteria. And you will see that many of these are inducible promoters. But does that mean that we never have a constitutive promoter for expression of protein in bacteria? No, in many cases, in many cases you use very strong constitutive promoters also, when you don't much worry about your protein being highly toxic to the cells, etc. And if the high expression of a protein is not affecting your this growth of your cells, So in those cases, you use very strong constitutive promoters as well. But in many times, what you use are the inducible promoters. So which are the inducible promoters that are generally used? One is LAGZ promoter, which you will find in many different vectors, commercial vectors. So what you have to do is you clone your gene downstream of this promoter, you provide IPTG from outside, and what you will get is the inducible expression of your protein. Then there is another gene called, promoter, tryptophan promoter. So this tryptophan promoter is highly regulatable. So if you add 3-beta indole acrylic acid, then it was seen that it brings about down regulation in the level of tryptophan in the cell. But it means it inhibits the conversion of indole to tryptophan somehow. So if the levels of tryptophan are low in the cell, what will happen is that it will lead to de-repression of the tryptophan promoter. So we will see high expression from this particular promoter of our gene of interest. And now if I want to, switch off this promoter, then what I will do, I will provide tryptophan from outside. So again, there will be no transcription. So this is how the tryptophan promoter based regulatable. So it's a regulatable promoter. You add certain chemical, it induces the transcription. You add another chemical, it decreases the transcription. So there is another promoter, TAC promoter, which is nothing but a hybrid of these two promoters. So what they have done, smart molecular biologists, they made a hybrid of this promoter. I don't know what was the reason they thought about it, but it was smart, means at least the results were very smart. Very good. So -10 region was from LAC and -35 from tryptophan promoter. So I hope that you know that these two are the sequences which are present in the promoter of bacterial genes. And so why when they made this hybrid promoter, what they found that the induction in the expression of these genes was manifold higher than the promoters, the original promoters. Let's say 3 times stronger than tryptophan and 5 to 10 times stronger than lactose. But also want to tell you that because this final promoter that is made, TAC or TRC promoter, it has the lac operator region. So this is inducible by IPTG only. Okay. So it is IPTG inducible promoter having these two regions, and these two names only differ in one base pair. So there is one TAC promoter which is separated where -10 and 35 are separated by 16 base pair, and TRC promoter are separated by 17 base pair. That's the only difference between the two. Okay, then there is this lambda PL promoter, which is affected by the presence of CI gene, if you remember. CI gene is that gene that I introduced you to when I was telling you about the phage vectors. So here what they have is they have cloned CI gene, which is temperature sensitive. So if the temperature is low, then CI gene will be made. If CI gene will be made, it will go and bind to this promoter and switch off the transcription. Now, if I transfer my bacterial cell to higher temperature, more than 30 degree, then what will happen? That even though the CI protein will be made, but it will be degraded because it is temperature sensitive. So if CI gene is not there, it will not go and bind to this promoter, and as a result, we will have high expression from this promoter. So otherwise, it's a very means strong promoter, but it can be regulated by this temperature difference. So in this case, you can see that if you want to induce the expression of your gene of interest, you have to add certain chemicals. But in this case, you don't have to use any extra chemicals. So from industrial point of view, it's very good. You just have to transfer your bacteria, which has your recombinant plasmid, from low to high temperature when you want to induce the expression. So that way, it is a very smart way of uh, making these inducible promoters. And the last is T7 promoter, which I've already told you. So T7 promoter is the promoter which you will find in many famous commercially available cloning vectors, and this is inducible in response to T7 RNA, means T7 RNA polymerase binds to this particular promoter, but this whole system is IPTG inducible. Why it is IPTG inducible? I will just come to that. So this is just your introduction to the various promoters which are present, which are being used for bacterial expression system. The yeast ones I had already told you before. So now coming to, I want to tell you in more detail the PET vectors, and where I will introduce you to the T7 promoter that I just told you in my last slide also. And let me explain to you in detail that what are these PET vectors. So these PET vectors are the vectors that are like very, very widely used for protein expression in majority of the labs worldwide which are working on protein expression. Those labs will definitely have the PET vector. So if you want to harvest your protein of interest, what you will do is that you will buy this PET vector and you will clone your gene downstream of the T7 promoter in the MCS region where you will clone your gene of interest. Okay, so this is the T7 promoter. Now, why T7 promoter and why not any promoter which is present from E. coli genome? Because it is very strong, very, very strong promoter. And but the problem is that it is only induced, means it is only recognized by T7 RNA polymerase and not by the RNA polymerase of the E. coli. Okay, so it means that the bacteria in which I am putting the PET vector, I have to somehow have high expression of T7 RNA polymerase as well. So what the engineers have done is that they have introduced that gene into the bacterial genome itself. So the host strains that are available for PET expression, they have this lysogen which is present in their bacterial genome, which has the T7 gene. It is staying there in the bacterial genome, and this gene, you know that it's from phage origin. So what they have done is that they have made this gene, or let's say cloned this gene downstream of a lac promoter. Okay, so it means that under normal condition, when IPTG is not there, then in E. coli gene, we know that we have LAC-I gene, which codes for LAC repressor. Please, you have to remember your whole notes about the LAC operon. So this will give rise to LAC repressor. This LAC repressor will go and bind to the operator region in the LAC promoter and inhibit the expression of the gene that is cloned downstream. Okay. Now in the cells, if I expose them to IPTG, what will happen? IPTG, which is a lactose analog, it will go and bind to this LAC repressor, and now LAC repressor will be no more able to bind to this LAC operator. If the LAC repressor is not bound here, what we will have that in response to IPTG treatment, the expression of T7 polymerase, RNA polymerase is very high. And this T7 RNA polymerase that is made, it goes and binds to this promoter, which is a T7 promoter, and induce the expression of our gene. To make it extremely, very, very specific in the expression, I mean highly regulatable in nature, apart from the IPTG induction based induction in this T7 gene, they have also added LAC operator in the T7 promoter. So apart from the IPTG induction of this gene, even this gene becomes IPTG induced. So it means it's double regulation in the levels, means that without IPTG it will be clean, no expression of the gene, there will be no basal expression, but in presence of IPTG there will be very high expression. Now in those scenarios in which the target gene is little toxic to the cell, what they have done is that they use a bacterial host strain in which, apart from this pet vector that you add from outside and this T7 gene which already is present in the host strain, they also put a small plasmid called P-lysis plasmid or lysi plasmid. So this plasmid, this I discussed with you when I was telling you about the E. coli strains and how they have been manipulated, or what extra features they have to have a regulated expression. So in this, those cases where target gene is little toxic, they add this particular plasmid in it. What it does, it induces the expression of T7 lysozyme, and once T7 lysozyme is made, it makes T7 RNA polymerase inactive. So it means that it will ensure that even though after IPTG we have very high T7 RNA polymerase which is being made in the cell, but to some extent it gets a little bit inhibited because of the some expression from P lysis of T7 lysozyme. So if T7 RNA polymerase is not being made to that high extent, so in those cases, the target gene will not be made to very, very high levels so as to make the whole bacteria, make it toxic for the bacterial cell. So this is the introduction to the pet vectors. How do you clone in pet vectors your fusion gene? There will be a separate class on that for as part of the practical or assignments that I've given you. So please go through these notes also, the Shine-Delgarno sequence, etc. So this is just one diagram of one expression vector. So here what we have is a promoter sequence. In this case, this is TAC promoter, a very strong promoter, which is induced in response to IPTG, because in the TAC promoter, they have added a lac operator sequence. Then there should be a ribosome binding site. So the transcript will be made from here, okay, and it will end here. For sure, it will end here. In many cases, let's say you clone your gene of interest here, and it could be that in your gene of interest itself, there may be a transcript termination sequence, depending on whether you have cloned your whole transcript or not. But in many cases, you just clone your coding sequence, which is giving rise to the protein. So you might not have your transcription termination sequence in your gene of interest. So in those cases, it will get terminated here. Let's say by some means it is not terminated here, then for sure it will be terminated here. So that's why two termination sites have been added. Okay, so your transcript is being made from the promoter region to here. But the ribosome binding site is here. So your protein will be made from here. And because your start site, I mean ATG site will be in your gene of interest. So the protein will be made right from here. And the ribosome will be able to bind here, which is close to it, and then initiate the translation here, and then your protein will be made. Okay. So that's all for today's class, and we will continue with the mammalian expression vectors and the fusion tags in my subsequent 1 or 2 class, and I'm open for questions.
Ma'am,
Huh?
till this class